All right, good morning. My name is Henry Childs. I'm a PhD candidate in the Donald Lab here at Duke University. I'll be talking today about DexDesign, which is our newest algorithm for designing de novo T-peptides. So this work was completed in collaboration with Nathan Warren, who is now graduated at 1063 Therapeutics, collaborator Pei Zhao, and also, of course, my advisor, Bruce Donald. We currently have the preprint available on bioRxiv if you'd like to check out this paper. Additionally, I spoke on this recently at RECOM at MIT, so, technical difficulties. At RECOM at MIT, so there's a copy of that as well in the proceedings. And then we recently were accepted for protein engineering design selection for a special series there, so keep tuned for that. So we designed all of these in something we call OSPREY, which is our open source protein redesign software. There's currently drugs and vaccines in 28 clinical trials. This includes an inhibitor for a trafficking protein involved in cystic fibrosis, a HIV-1 antibody that's highly potent in vitro and in vivo, and many others. So I recommend checking out these papers. I think they're very interesting. That being said, these are mostly canonical design. I'll be talking about something a little bit differently today. So this talk is going to be in two pieces. I'm first going to have a background. I'm going to talk about D-peptides and our software suite OSPREY. I'd like to jump into the new algorithms, but first I need to cover some basics and some of the background, and then I'll be jumping into our new algorithm, DexDesign, and those methods and results. So first off, let's talk about L-peptides, what exactly they are. So here we're seeing PDB-ID 6OV7. That was that aforementioned inhibitory peptide for that trafficking protein in cystic fibrosis, which was designed using OSPREY in the Donald Lab. These have a number of benefits. These include very high specificity, really good efficacy. Since the 1921 synthesis of insulin, over 60 of these have entered FDA approval, and they also have very low immunogenicity. That being said, despite these advantages, there's also a number of disadvantages to peptides. Specifically L-peptides, that includes poor bioavailability and short retention. So the question is, we know that these peptides are really beneficial therapeutics, but how do we have the advantages while avoiding the disadvantages? One of the ways to do that is with D-peptides. So specifically, when I say D-peptides, I'm talking about non-canonical amino acids. So L is Latin for levo, meaning left, and D is dexter, is Latin, meaning right. So there's a number of things to know about this diagram. Here on the left, we have our L-peptide. Here on the right, we have our D-peptide. The first is that this is a reflection operation, so without loss of generality, we're showing this L-peptide being reflected across the XY plane. Note that even for amino acids with multiple stereocenters, such as threonine, this inverts all the stereocenters, so 3R goes to 3S, and 2S goes to 2R. Also note that reflection is an energy equivariant geometric transformation. This means that this reflection corresponds to a symmetry in the energy field of the protein. So this doesn't change the energy of the structure, it only inverts the fold. So this will be important moving forward. So you might ask, okay, this is cool, you've outlined L-peptides, you're seeing a D-peptide, why does this matter? So there's actually a number of advantages to D-peptides. The one I'll be talking about today is the fact that it can actually account for some of the drawbacks of L-peptides by evading protease recognition. Specifically for something like serine proteases, it's been shown that D-peptides are destabilized in the active site of chymotrypsin and last much longer in the body. So we designed all of our designs, specifically I'll be talking about D-peptides, in OSPREY. That stands for Open Source Protein Redesign for You. And we actually frame protein design as an optimization problem. So how do we actually solve for some of these structures and calculate our predicted binding affinity? So you can think of this as designing a protein energy landscape. So we have our energy, which is a function of our conformation, which we want to enumerate very efficiently. So one of the ways we can do that is define a GMEC. A GMEC stands for the global minimum energy conformation. It's basically the conformation is the shape of the protein that gives us our minimum energy. So I'll be talking a lot today about finding a GMEC. It's a lot simpler conceptually in the talk to talk about finding one minimum energy. But do note that in reality, when we are predicting a binding affinity, we actually use an ensemble method, which allows us to sample from many energy minima and many different energy wells, which is reflective of how dynamic and flexible proteins can be. So how do we do this in OSPREY? Short answer, it's really complicated. A simplified answer, we can do it in 4 steps. So the first is our input. We have input, which is our biophysical model. This is a experimentally determined protein crystal structure. Second, we have an energy function. So OSPREY actually uses 2 different energy functions. We use EF1 for an implicit solvation model, and we also use AMBER tools for our van der Waals and electrostatics. Third, we also have our sequence space, so this is the mutations we're checking for in the structure. And lastly, we also have our flexibility, which we use continuous flexibility in OSPREY. From there, we prune using something called dead-end elimination, which I think is really exciting. I'm happy to share that with you all today. Third, we then enumerate these energy intervals to construct a confirmation space using the OSPREY algorithm, which is a graph search. And then finally, once we do all those calculations, we want to do something with it. And in this case, we're going to be looking at binding affinity. So let's first, once we have our input and we've specified our design space, let's look at dead-end elimination. So first, let's look at discrete dead-end elimination, and then I'll move on to continuous. This is conceptually a little bit more easy to understand, a little bit more digestible. So we're guaranteed not to prove that GMAC. Remember, that's on the graph. We're guaranteed not to prove that GMAC using this dead-end elimination. Let's talk about how that works with a toy example. So we have here our protein in gray. It's an example protein, and we're trying to decide if we want to choose rotamer R or rotamer T at position I. And we want to determine that by looking at the intra-residue energy, as well as the pairwise interaction energy of rotamer S at position J. So I should note, I'm only showing one rotamer here for simplicity, but this is actually dynamically determined, so this can change to any of the rotamers at position J. So the easiest way to understand this is to look at it in terms of left-hand and right-hand side. So on the left-hand side, we have the intra-residue energy of rotamer R at position I, and then we're looking at the minimum energy interaction between rotamer R and rotamer S between position I and position J. Contrast that with the right-hand side of this equation. We're looking at the intra-residue energy of rotamer T, and also its maximum interaction between rotamer T and rotamer S. What this means is the best case energy for rotamer R at a higher energy than the worst case energy with rotamer T, and if so, we can prune that from our confirmation space. This is when we can limit the amount of confirmations that we have to enumerate. So this is discrete. If we do it with continuous flexibility, it ends up being a little bit more difficult. We change our parameters a little bit. So we use something called voxels. I typically say pixels are to 2D, as voxels are to 3D. They're a way to segment 3D space. So you can imagine taking the chi1, chi2, using cyclic coordinate descent or some sort of gradient descent algorithm in order to model continuous flexibility. We're bounding, I'm showing here, these amino acids within a voxel. This is a little bit more difficult to parameterize, so we end up adding two additional considerations. We have first this optimistic lower bound. Basically, if we remove the steric constraints from the amino acid, what can it optimistically minimize to within that voxel? And then we also have an optimistic, we call this an interval energy, and this is saying once we reintroduce their constraints, how much of the energy of that rotamer, how much will it increase? So this is a little bit complicated, but basically all this means is if we minimize, is our minimized best case at a higher energy than our minimized worst case? If so, we can again prune that rotamer. So now we have a reduced confirmation space that we're going to explore. We do that using the A-star algorithm. So we're enumerating over energy intervals using the A-star algorithm. So you can look at our cost function here. So we have f of n, which is our cost function. For a node, each of these nodes is actually a sub-confirmation space. And that's going to be the energy of the current node, and then we have our h function, which are energy to the next node. And because we've already ordered the confirmation space using dead-end elimination, this is an admissible heuristic. So finally, we've done all this calculation, we've enumerated this confirmation space, and we actually want to calculate binding affinity. We use something called the K-star algorithm. So to approximate binding affinity, we first build a partition function for a given state. This is X. This can be our protein ligand or our protein ligand complex for a given sequence. And then we sum all the confirmations in that confirmation space, and then we Boltzmann weight them here with the energy of our confirmation over RT. We build partition functions for our protein ligand complex. We take the ratio, and that gives us our approximation of binding affinity. So a lot of you will probably recognize this similarity to Ka. Visualization of this, we're taking an ensemble of our complex, divided by the ensemble of our protein, ensemble of our ligand, constructing these partition functions. And this gives us an approximation to Ka. So that's a little bit of background about how we're enumerating these confirmation spaces in OSPREY and how we're doing binding affinity. I'm now going to jump over to DexDesign. So the question was, we have all of these great tools in OSPREY, but how do we actually do de novo design? This is really difficult. So there's a whole bucket list of reasons why this is really difficult to do. The first that I'll highlight is data scarcity. So a lot of people ask, why don't you just do machine learning? There's a lot of scarcity of structural data of deep peptides and complex with L proteins. So it wasn't really an option to do this in DexDesign. We hypothesized, though, if we use the fact that reflections are energy equivariant, if we used a substructure search, which we'll talk about a little bit later, and if we also implemented novel de novo design methods, we could probably design some deep peptides that are predicted to bind with L proteins. So let's walk through this DexDesign algorithm. So step one of DexDesign. select an L inhibitor and L target. Again, this is that PDB-ID 60V7 from the beginning of the presentation, KCAL01. We specifically selected this. We had published on this system previously. We understood where the hydrogen bonds were interacting. We understood structure was going on. It would give us some evaluation metrics after we did some of these designs. So we selected this L inhibitor in combination with this L target. We mirrored it to D space. So now we have a D peptide in complex with a D protein. Third, we then separate out the components. So we're going to design a D peptide. So let's ignore DCalP for now, and we're going to get DKCal01. Now this comes our substructure search. We use something called MASTER from the Gregorian lab. This stands for Method of Accelerated Search for Tertiary Ensemble Representatives. What this means is it's a geometric hashing algorithm. We're giving our database of over 119,000 crystal structures our query, which is DKCal01. And what it's doing is it's aligning the alpha carbons in the backbone of DKCal01 to structures in our database. And it's returning L peptides that look like our query of DKCal01. So this is how we're generating a synthetic structural data set using a substructure search. And these are all mined, high quality, I believe 2.5 angstroms and lower crystal structures from the PDB. So once we have a synthetic data set, we then align all those L structures back into the complex of DCalP, where we do one final reflection operation. We do our binding affinity K star, and then we now have a D peptide, or D design, in complex with alpha-P. So that's how we are generating some of these designs. So that's an overview of DexDesign. Once again, starting with L peptide in complex with an L protein, reflection operation, extracting out our D peptide. We are doing a substructure search to generate L peptides. We then align and reflect and do K-star algorithm. So some of you all might have noticed the K-star algorithm, we talked about how computationally complex some of these things are. This is by far in this lab set the most computationally complex. It's really, really difficult to do is actually taking substructures from the PDB, aligning them, and just saying that they're going to bind. It's actually hard. We have to do some designs. We can't just take that wild type sequence. So in order to tackle this computational complexity, it's intractable if you want to just enumerate the whole conformation space, and sequence space scales exponentially. We implemented 3 techniques. We call the minimum flexible set, inverse alanine scanning, and then the K-star-based mutational scan. So first, the minimum flexible set. This is step 1 of this redesign technique. So what MFS is essentially doing is updating scaffolds. So these scaffolds are substructures from master to new chemical contexts by locally ideal global geometry. So you can think about doing dead elimination, continuous, on these substructures in order to update their scaffolds to their new targets. So what we do as step 1 is we prune aligned substructures with too many unfavorable steric clashes. Typically, these are computationally intractable. And then we update these using our algorithms to the new geometry. So an example here, on the left we have a D peptide from master in complex with an L protein. So in cyan is our D peptide, in green is our L protein. And what we're looking at here, this is from the Richmond lab probe dots. We have an implementation in the Donald lab called our protein design plugin. So we're visualizing here, so we have purple, red, and yellow are all visualizing bad and small steric overlap clashes. We can see here with this sidechain. And then we use some of our algorithms, update the geometry, and to resolve some of the steric clashes. So we're doing more realistic modeling at a lower energy. From there, we actually want to start changing the sequence. How do we actually optimize our binding affinity given the substructure? So the second we call inverse alanine scanning. So inverse alanine does 2 things. It gives us an optimistic upper bound in our increase in K-star score. By the way, increase in K-star score indicates an increase in predicted binding affinity. And it also gives us our maximum flexible set for point mutations. Before I talk about exactly what that means, let me talk about what this is based on. This is inspired by the technique of alanine scanning, which is typically experimental. Typically, in experimental alanine scanning, you'll mutate one residue to alanine and measure the change in the change of binding. We typically are called hotspots. So if you mutate a residue to alanine and then a place binding, you understand that residue is very important for binding. In contrast, in inverse alanine scanning, which is our proposed computational technique, we mutate all residues to alanine except one. And that one residue is checked at all rotor identities at all of the amino acids. So what this is doing is it's looking at if you give this ligand optimal geometric constraints, so you're removing a lot of the steric constraints on the neighboring residues. We also allow translation rotation of ligand in that pocket. What's the maximum binding affinity contribution that one residue can make given any amino acid affinity? So an example here, we're looking again at the difference between our mutant and our wild type, and we're allowing translation rotation. Notice the difference in this backbone. In this case, we're just sticking with the wild type. We found a different rotor identity that's predicted increased binding affinity. We can also change the sequence as well. Last but not least, We have our K-star-based mutational scan, which gives us a constraint upper bound on our increase in K-star score, and again, a maximum flexible set for simultaneous mutations. So in our K-star-based mutational scan, we iteratively mutate all residues. We iteratively mutate each residue to all 20 amino acids, and then we evaluate for large changes and increases in binding affinity given multiple simultaneous mutations. So here's an example here. So we have, again, our D peptide comes with L protein. We commonly saw that this increase in K-star score from 22 to 26.1 on a log 10 scale. So using all these methods, updating the geometry, changing the sequence with inverse aligning scanning and K-star-based mutational scanning, particularly give us D peptides that are predicted to be very tight binders to L proteins. So here's an example. Again, our D peptide in cyan, L protein in green. So let's think in terms of left and right-hand side. So on the right-hand side, these are the things, think back to step 1 of DexDesign, we selected a known system that we well understood, is we're looking at beta sheet extensions here in panel A. In panel D, there's hydrogen bonds that are predicted to be established with the carboxyl binding loop. This is mentioned in previous literature as being important for very high binding affinity. We've also commonly noticed that our D peptides were restituting novel interactions not present to the L protein. So notice here, for example, we're establishing new hydrogen bonds given the unique geometry and inverted fold of our D peptide, and also having favorable van der Waals context as well. So let's look at some results. This is for KCAL01. At the top here, we have our CFTRC term. This is the endogenous ligand. We have our, in red and green, we have our predicted hydrogen bonds. And blue, we have our predicted binding affinity. Note that all of our designs, this is CalP-Pep15 through CalP-Pep9, are all predicted to outcompete the endogenous ligand, which is the primary design objective of an inhibitory peptide. We are commonly recapitulating and restituting favorable hydrogen bonds and interactions. Down here at the bottom is actually KCAL01 to the starting structure. We're not predicted to outcompete that binding affinity. That being said, as I mentioned at the beginning, there's many advantages to D peptides that could overcome this predicted lack in binding affinity. So that concludes both sections of my talk. I'd like to thank members of the Donald for their very helpful discussion. If you're interested in anything up here today, I'm happy to talk. My email is henry.childs@duke.edu. It's mentioning all of this software is completely free. It's on our GitHub page. We also, it wasn't mentioned on the first slide, we just submitted this week a protocol paper we invited to submit for JCB, which goes through this entire procedure step by step. It gives you video tutorials that are recorded over an hour of video tutorials about how to use DexDesign, includes commands, set up protocols, everything. And if you want that before it's published, I'm happy to send that to you or talk and provide guidance. So thank you all so much, and if I have time, I'll open for questions.
Yeah. We've got a couple